Hello guys, this is Malinki. Welcome back to my channel Voice of Malinki. So today we will discuss about inflammation and fever. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. Let's discuss inflammation and fever. So inflammation refers to our body's fighting process. So it is our body's fighting process against microbes to heal itself. So inflammation is our body's fighting process. So our body fights against microbes to heal itself, right? So this is inflammation. And inflammation is a component of innate immunity. So inflammation is a component of innate immunity, and when we have a tissue injury such as a cut, microbes get opportunity to enter our body. Right. So this we all know that if we have a tissue injury such as a cut, so microbes can enter our body from outside. Now our immune cells are targeted to this site. So suppose there is a cut and our immune cells will be targeted to the cut site and they destroy these microbes at that site so that microbes don't get opportunity to spread further. Right? Okay. So suppose this is a cut and then our immune cells, they will go toward that side, because some bacteria or some other microbes could enter through this cut. Now our immune cells will go toward this site of infection and it will destroy those harmful microbes so that those microbes will not get any opportunity to spread further. So this is inflammation. Now the signs and symptoms of inflammation. So 5 signs and symptoms of inflammations are there. First is the tissue redness. So tissue becomes red, and then heat generation. So we sometimes feel the heat because it is warm. The place is warm, the tissue is warm, we can feel it. And then swelling of the tissue. That also we have seen after a cut or after an injury, we have seen some swelling part of the tissue. Then there will be some pain and the loss of tissue function. So these are some signs and symptoms of inflammation. Next we will start discussing about the mechanism. So after a cut, microbes enter our body. So after a cut, microbes enter our body and resident macrophages of that tissue identify the invading microbes of that particular site. Okay, so suppose this is the cut and the bacteria or some microbes are entering our body. So some are entering our body, these are some microbes. And at that time, the resident macrophages means those macrophages that are residing in that particular tissue. So suppose this is the skin and these macrophages are skin dwellers. So they are found only in the skin. They are some resident macrophages, right? So what they will do, they will identify, they will detect those microbes. So these resident macrophages will identify and detect those microbes. And how does this interaction or detection occur? So interaction between macrophage and microbe occurs by PRR, that is present on the macrophage. So PRR is the pathogen recognition receptor. So we know that what is PRR? It is the pathogen recognition receptor. So these these are some receptors that are found in the membrane, plasma membrane of macrophages. And PAM. So PAM is present on the microbe. So PAMs are pathogen associated molecular pattern. Okay, so these are some substances that are found in the microbes, and the interaction between microbes and macrophages occurs via these two receptors like PRR and PAMs. After the interaction, resident macrophages are activated. So when the interaction occurs between PRR and PAM, or you can say between macrophages and microbes, right? Then the resident macrophages are activated. So then the resident macrophages of the particular tissue will be activated and they will release some pro-inflammatory molecules. So they release some pro-inflammatory molecules. Those pro-inflammatory molecules are interference alpha, interleukin 1, etc. Okay, so these are some pro-inflammatory molecules. These pro-inflammatory molecules will be released by those macrophages. Those are now activated by the microbes. Okay. Next is apart from them means the TNF alpha and interleukin 1. So apart from them, one more pro-inflammatory molecule, that is kinin. So it is normally present in the blood plasma in an inactivated form. So when there is no any infection, this kinin is present in our blood plasma in an inactivated form. But tissue injury activates them. So tissue injury activates this kinin and a particular kinin. So kinin are of different types. So especially a particular kinin that is named as bradykinin, it stimulates pain receptors in the skin. So the bradykinin, what does it do? It actually stimulates the pain receptors in our skin and it causes pain. So that's why after an tissue injury, we feel some pain because these bradykinins are those substances that can cause pain. And in generally, these kinin or bradykinin, they are found in our blood in the inactivated form. But when an infection occurs, these bradykinins are getting activated and then they will cause some pain. And pain normally cause an individual to protect the injured area. So pain is beneficial in this case. Pain is beneficial because it can protect the individual. So it can protect the injured areas of the individual. So pain is beneficial. Now next is the pro-inflammatory molecules. So some pro-inflammatory molecules that I have already told, those are the TNF alpha and interleukin 1. So these pro-inflammatory molecules trigger vasodilation and capillary permeability. So these pro-inflammatory molecules do two things. One is the vasodilation and two is the capillary permeability. So what is vasodilation and what is capillary permeability? So vasodilation is a process. Vasodilation is a process where the diameter of blood vessels increase. So the diameter of blood vessels increase. Suppose this is the normal blood. This is the normal blood vessel, right? Normal blood vessel. And these inflammatory molecules will act on them. And then what it will do, it will increase the diameter. That means now the blood vessel becomes wider, right? Blood vessels become wider. So this thing is done by the pro-inflammatory molecules and this is called as vasodilation. So vasodilation induces increased blood flow to the damaged area. Because when the blood vessel is wider, it is wider, so definitely, obviously, it will increase the blood flow to the damaged area and it causes tissue redness and heat. And as the blood flow is increased, definitely it will cause the redness. It will cause the redness and it will cause the heat. because the blood flow is now increased. So this is vasodilation. And next is capillary permeability is also increased by those pro-inflammatory molecules. So an increase in capillary permeability facilitates the influx of fluid and cells from capillaries into the site of infection. So that means if these are the blood capillary, so they will allow the influx of some fluid and cells from these capillaries into the site of infection. Suppose this is the site of infection. And this is called as capillary permeability because the capillary is now permitting something. The capillary is now permitting some cells, some immune cells. So capillary is now permitting those immune cells to reach the site of infection from the blood. So if these are the immune cells, these are some immune cells. So now capillary will permit them to cross the blood capillary and to enter the site of infection. So immune cells now, these are now reaching at the site of infection. And not only the cells, definitely some fluid will also be there in the site of infection due to the capillary permeability. And this fluid accumulation contributes to tissue swelling, right? So now this fluid accumulation is causing the tissue swelling because lots and lots of fluid is now coming to the site of infection. That will contribute to the tissue swelling. So I already have told that the cells are now permeable, the cells are now permitted to reach the site of infection. And among those, mainly phagocytes are the cells. Phagocytes or phagocytic cells, they are the main cell that will enter the site of infection due to the capillary permeability. So they will now reach to the site of infection. And among those phagocytes, neutrophils are the first cell. So please try to remember this thing always because many times question can come like that which cell is the first cell that reach the site of infection. So neutrophil is the first cell that reach at the site of infection and then followed by monocytes and other. So first is the neutrophil that reach at the site of infection and then second is the monocyte. Then after neutrophil, monocytes will reach at the site of infection and then other phagocytic cells will also reach at the site of infection. Okay, now the immigration of phagocytes from the blood capillary to the site of infection is a multi-step process. So how the cells or phagocytic cells or phagocytes, they reach at the site of infection from the blood capillary, it is not a single step process, rather it is a multi-step process. So we will now see that process. So first the phagocytes adhere to the wall of blood capillary. This is called as margination. So suppose this is the blood capillary. This is the blood capillary. And some phagocytes are there. These phagocytes are flowing in the blood capillary. And first of all, they will adhere. So they are now adhering to the wall of blood capillary because this is the wall of blood capillary, right? And this phagocytic cell, this phagocyte is now adhering to the wall of blood capillary. And this is called as margination, right? This is called as margination because the phagocytic cell is now at the margin of the blood capillary. So this is the first step. Now second step is they pass through the capillary wall. So this is called diapedesis or extravasation. So first of all, they are now these phagocytes are now adhering to the side, to the wall of the capillary. So this is the wall of capillary. Now what will happen? This is the phagocytic cell that will now pass through the capillary wall. So the phagocytic cell is now passing through this capillary wall. So this is called as diapedesis or extravasation. Finally, they migrate to the site of infection. So finally, this is the site of infection and the phagocyte has reached there. So this is the phagocyte. This is the phagocyte and it has reached in that particular infection site. Okay. So this is called chemotaxis. So this is called as chemotaxis. Okay. Now, as phagocytes accumulate at the site of infection, they begin to phagocytose microbes because phagocytes are now there at the site of infection. So this is the site of infection, and in that site you will get number of microbes like that. So what they will do? Phagocytes undergo phagocytosis, we all know. So now when the phagocytes have reached their site of infection, they will start phagocytosis. They will start phagocytosis and then they will kill those, they will destroy those microbes. They will engulf those microbes and then they will release some lytic enzymes. So these phagocytes will now release some lytic enzymes. So the phagocytes are now undergoing phagocytosis and it will release some lytic enzymes. And those lytic enzymes can damage and kill the nearby healthy cells. So in the spot or in the site of infection, other healthy cells are also there, right? So in the site of infection, phagocytes are there, some microbes are there, and some healthy cells are also there. And the release of these lytic enzymes will kill those healthy cells also. And the accumulation of dead cells, phagocytosed microbes and fluid forms a substance that is called as pus. So phagocytosis has been done and some microbes are now digested. So those microbes and those dead cells and the fluid, so they all will form a substance called pus, and it is a thick yellowish fluid. So pus is a thick yellowish fluid that is now formed. And vasodilation and increase in capillary permeability in an injured tissue also enable enzymes of blood clotting system to enter the tissue. Because we know that vasodilation occurred, capillary permeability increased. That's why they are allowing some cells, they are allowing some fluid to enter the site of infection from the blood capillary. And when they are allowing these cells and these fluids to go to the site of infection, they will also allow some enzymes of blood clotting system to enter the tissue. So enzymes of blood clotting systems will also enter the tissue, that means the site of infection. These enzymes promote the deposition of fibrin. So now the enzymes of blood clotting system are there in the site of infection and they will promote the deposition of fibrin. And what is fibrin? Fibrin is the main component of a blood clot. So fibrin is the main component of blood clot. So what will happen? The fibril strands wall off the injured area from the rest of the body and serve to prevent the spread of infection, because now blood clotting will be formed. So now blood clotting will be formed because these fibrins will now form the blood clot, and the blood clot means the wall off. It will actually wall off the injured area from the rest of the body, and so that they can prevent the spread of the infection anymore. Okay. So next is inflammation is also associated with fever. So we know when inflammation occurs, we have some fever also. So how does fever occur? Now, pro-inflammatory molecules, that is TNF alpha and interleukin 1, So these are some pro-inflammatory molecules that I have already mentioned, that these are released from the tissue. These are released from the tissue macrophages, and what they will do, they are actually generated after an infection, and they are also called as endogenous pyrogen. So we have seen that they do the vasodilation and increase in capillary permeability. But other than these, they are also endogenous pyrogen. That means they act on hypothalamus. And we know that hypothalamus is a central portion of our brain that can control our body temperature. So it controls our body temperature, and these pro-inflammatory molecules will act on it and cause a rise in body temperature or fever. So in this way, fever is formed after inflammation. Now fever is beneficial. So sometimes fever is beneficial. And why fever is beneficial? Because it helps immune cells to crawl along blood capillary wall to attack invading microbes. So the fever, the high temperature, will allow the immune cells to crawl along blood capillary wall to attack invading microbes. And most of the microbes do well when our body is at normal temperature. But it is harder for them to survive in high body temperature. In this way, fever can inhibit the growth of microbes, because microbes can't grow at high body temperature. And once most of the microbes have been cleared, so when most of the microbes have been cleared, how they were cleared? They were cleared by phagocytes. So they were cleared by the process of phagocytosis, and then tissue repair and regeneration of new tissue will begin. So now the tissue repair and regeneration of new tissue will begin. Okay, so we will see the whole thing in very briefly in this picture. So here you can see the tissue damage is occurred, and then when the tissue is damaged, so these bacteria can enter the tissue, so they can enter our body, and then the tissue damage causes release of pro-inflammatory molecules. So it causes the release of some pro-inflammatory molecules, and release of pro-inflammatory molecules by what? By resident macrophages. And these pro-inflammatory molecules means TNF alpha, interleukin 1, and they will trigger vasodilation. They will trigger vasodilation and increase in capillary permeability. Okay, now permeable capillaries allow an influx of fluid and cells. So permeability allows them influx of fluid and cell from the blood capillary into the site of infection. And in this process, the first step is the margination, and then is the extravasation, and third is the chemotaxis. And when these phagocytes are in the site of infection, so they are now migrating to the site of infection, so they will destroy bacteria by the process of phagocytosis. So this is all about the inflammation and fever.